Arjun. Arjun also presented, I think. So what was Arjun's idea?
We have tried for genome edited breeding for dry rice are ready basmati varieties because
ये
the
पॉइंटर
major
है यहाँ से
issue with basmati lines with these days are they are losing their aroma. If we are planting it
ठीक है
at
ना?
अच्छा ये पॉइंटर है, ओके, और ये
so it is
नेक्स्ट का
being
है, ठीक है।
governed by a gene which is bad H2 and bad H2 is a negative regulator of 280 which is 280 so if we are going to knock out that particular bad H2 gene we are going to promote aerobic content in the rice. That was our idea.
Okay, good. Again in rice. So 2 rice, 1 banana, and now we are going to soybean.
यस सर,
Okay, please go ahead. The Nakul team,
यस सर।
the stage is all yours.
Good afternoon, one and all. I am Khushbu and from Group 4, and we are going to present on the topic genome editing through CRISPR-Cas9 for improvement of oil quality in soybean, that is glycine max. So, soybean is an essential oil seed plant which produce plant-based protein that make is very crucial and it's very crucial plant for make it very valuable and crucial plant. So, if we talk about the content of soya bean seed is majorly contain around 52% of linoleic acid, while it contain only 25% of oleic acid. So if you see the pie chart which is present on your right hand side, which represent the soybean production in different state of India, which is majorly cultivated in Madhya Pradesh. So the question is, why do we need to improve soybean oleic acid in soybean oil? So the reason is the oleic acid is a monounsaturated fatty acid, which is essential to improve its oil quality and which is very essential for consumer. And second one is oleic acid increase the oxidative stability of oil, which finally increase its shelf life and make it suitable for preparing food.
While considering all the importance and area regarding, after that we chosen some problem regarding this soybean. There is one major problem, that is the oil, which is oleic acid. For improvement of this oleic acid, we have to many more improvement state from traditional way. We also improve some like that crossing plant, after the selecting the crossing some offspring, and after that offspring we do get a desired gene which is inserted in a genetic material. But for this traditional method is for changing the whole genome or some genome, and this traditional and mutagenesis technique is only which is safe for us, and which is not unregulated by any regulation committee, because this is not harmful or somewhat not any harmful effect on us. So next, that is the after that mutagenesis, one of the technique we can improve some our target by exposing seeds to some various mutagen, that is some physical mutagens and some are the chemical mutagen. By using this mutagen, we can use get our some random changes in your unpredictable random changes, but these are unpredictable. So that's the gene affected due to this is much more, that is up to 1000 or up to 10,000. So this is the method, but in this method there is no precise change is there. Here only the general random or unpredictable change, so that's why we don't get that much effective result. So after After we get this DNA, that is Watson and Crick model. After that we go towards the some improved technology, that is the RNAi transgenic technology, and last one, that is the genome editing. In RNAi we just some target genes are somewhat switch or some silence, which is relevance up to the one or up to the dozen of genotypes, which is safety, but which is regulated by some committee, which is in this technique there is a delivery method is up to the limited servers. After that, the transgenic, in that the inserting the selected genes using the some DNA methods, recombinant DNA methods. In that we go only for the some up to the 8 genotypes we have to affected due to this training, due to this improvement strategy, and which may some adverse effect. So that's why it's somewhat regulated by some institute also. And after that we go towards, this is a new and most precise editing technology, that is the genome editing, which is used to delete some gene, or somewhat we have to add also, but through the regulation committee we only he allow us to somewhat changing homologous or only you can somewhat delete some this gene. And in these techniques, which is very precise one and somewhat less consuming, less time consuming, so this was one of the beneficial. So we go forward this through this.
So let me gather your focus towards the gene of interest, that is FAD2, and the gene are like GMFAD2A and GMFA22B genes, which are located on chromosome 10. And so the focus is to basically upregulate the production of oleic acid, which is beneficial not only towards the health point of view, but also in production of biodiesels. Since I am from a biological science background, life science, I'd like to indicate that consumption of oleic acid is extremely important in a country like India, because we are predisposed to diabetes type 2 and increased heart risks. So this particular fatty acid is known to actually protect us from especially the insulin resistance case, that is type 2 diabetes. So what here we are looking forward to do is basically knock out or mutate this FAD2 enzyme, that is fatty acid desaturase 2, which actually causes the desaturation of oleic acid and converts it to linoleic acid, which is again polyunsaturated acid, which is harmful for heart health. And with this I think we are going towards the gene editing portion of our slide.
First of all, design the guide RNA and synthesis. First of all, fatty acid FAD2 gene sequencing download in NCBI, and primer design PCR amplification FAD2 gene, and PCR product purification and sequencing analyzed by chop method, and designed to guide RNA. Strategy for genome editing and guide RNA system. First of all, design the guide RNA and cloning to binary vector, and transform agrobacterium transform in plant, and screening the transformation plant, and PCR and aridization genotype, and selection the genetically edited plant with modification, and design the guide RNA via chop method. It is construct the CRISPR-Cas9 plasmid, this PE201B. Thank you.
coming to work plan, firstly, we are going to select the variety for doing fatty acid profiling, and in second step we are going to do fatty acid profiling of the genotypes, and for that we are going to do with GCMS, that is gas chromatography mass spectrometry, and also we are going to determine the oleic by linoleic acid ratio, and comparing the genotypes, we are going to select the cultivar which are going to edit, and in the third step we are going to standardize the protocol for the selected variety through use of various plant growth regulators and explants. And in the fourth step, for designing the guide RNA, first we are going for sequencing through Sanger sequencing. As Ashish already told we are going to design the guide RNA. That is, we use primers from the data obtained through NCBI, and we will give for sequencing. And after obtaining the data from sequencing, we will use bioinformatic tools for designing the guide RNA like CRISPR and ChOP. And we will, after designing, we will send for synthesis of guide RNA for outsourcing, and for construction, and then we will go for construction of Cas9 and guide RNA into a dicot vector, and then transformation to E. coli. For that, ligation of guide RNA was done by T4 ligase, and then cloning of vector was done to E. coli by heat shock method, and validation of the conformation was done through PCR and restriction digestion, and subcloning of these vector to agrobacterium was done through electroporation. And in step 6, we are going to transformation through co-culturing of agrobacterium with soybean explant. For this, agrobacterium mediated transformation was selected because agrobacterium is easy, feasible, and have high transgene delivery. And in the next step, we are going to select and regeneration of transgenic soybean plants by growing the explant in selective medium, and whole plant is regenerated through tissue culture techniques, and genotyping was done by PCR based conformation like indels and caps, and sequence was also confirmed by sequencing method. Thank you.
Okay, thank you. Any questions for the Nakul group? One question. Have you come across during your literature survey that something like this, I mean, high oleic acid soybean varieties, have they been released in some country? So can you elaborate on that? One can tell.
Sir, I think it is released, but it's outside India, so that's why I'm guessing introducing in India for high yielding varieties.
Problem, it's okay. But have you come across where specific and what kind of that variety is,
Not
gene and all? Okay. So I think in US it has been released, the high oleic acid soybean variety. Any other questions for the Nakul group? If not, let's have a round of applause, and all of you can come forward.
Thank you, sir.
For a good photograph of the Nakul group.
Thank you, sir. Yeah, then we'll just go towards our first slide.
आजकल तो एआई से कोई भी फोटो लग जाता है, मेरा तो लगा दिया एआई से फोटो। इस तरह से तो नहीं हुआ है, जिससे आप खिंचवाए वैसे फोटोग्राफ हुआ है। अच्छा, वो सारे टीम मेंबर्स नहीं थे उसमें। हाँ बस, if you want you can come here for the photograph. जल्दी आइए, जल्दी जनरेशन है। वही कॉपी करनी है उसकी, ले लीजिए। लीगल एस्पेक्ट के लिए ओके, स्टॉप ही कर दिया उन्होंने।
ये है सर।
इससे बोलेंगे तो जाएगी यूट्यूब में. ओके. हेलो, इट्स गोइंग.
लेकिन सर यहाँ पर ये साउंड कम जाएगा.
इससे जा रही है यूट्यूब पे.
यूट्यूब पे तो जा रही है, जूम पे
ओके, जूम का अलग चल रहा है.
अलग चल रहा है. आपको यहाँ पर
Okay. Usually I roam around in my classrooms rather than tied down at a single place. But it seems that this mic is very important for our Zoom participants. So I will be sticking on this one. So one good thing has happened is that Dr. Bhattacharya has gave his presentation on SOPs, Standard Operating Procedures. So this lecture, the intellectual property landscape and legal aspects of genome editing in plants, this lecture is designed to tell you about the overall legal aspects of doing genome editing. So in that, SOP is one of the part of that, which of course we are going to skip in this presentation. And maybe in next 25 minutes or so, I will go through some of the slides, but would like to give you some idea about the intellectual property landscape. So you have, for the last 1 week, you have seen many different aspects of genome editing, the science behind it, the applications behind it, right? And you know, everybody is like very excited to use genome editing in their work. But today, with one presentation, the SOP presentation, you must have got some hint that it is not so easy also, because after all, the products of genome editing is essentially the products of recombinant DNA technology. So all the things which are applicable with recombinant DNA technology are also applicable with genome editing. And therefore, it also increases any product development pipeline. But there is another element to it, and that element is the intellectual property. So we are going to touch on those aspects today. So what these buildings have to do with genome editing? So it's our new parliament building and different legislatures of different countries, Supreme Court. So it's genome editing we are not doing only in the laboratories, and genome editing is something we are discussing not only in a setting like this, but genome editing, recombinant DNA, biotechnology are also discussed in these buildings, in these places. So they are making different kinds of laws which affect the research ecosystem. So when you will enter into this research ecosystem, many of you are PhD students, probably many of you will join the private sector and will lead different departments of many companies. Then these issues will come to you on a almost daily basis, the regulatory aspect, the legal aspects. So as far as India is concerned, so I'm talking only specific to India, but also give you some global hints there, but will be mostly for India. So these 11 laws, they are very relevant to biotechnology. So anybody who is having a biotechnology program must know about these 11 laws. I will quickly just tell the names, but we'll stick to only the Patents Act. So Destructive Insects and Pests Act 1914. So this is an act of 1914. There was no genome editing at that point of time. Even genetic engineering was not there. The science of molecular biology was not there. But this is one law which affects the different biotech crops, including the genome edited crops. The Seed Act 1966, Insecticides Act 1968. So what does this Destructive Insects and Pests Act? Because I am not going into these laws in my classes, I go for each of them. So what does this law say? Destructive Insects and Pests Act 1914. At least agriculture students should be able to tell. पिछले टॉक में भी थोड़ा सा हिंट मिला था. So this law lays down the quarantine regulations of our country. right? So import and export of germplasm, because anyway any crop improvement program is about germplasm. One has to develop germplasm. Germplasm is imported, exported. Probably I have one slide of showing one example also. So all this import and export of germplasm. In one of the slide you have seen that any exogenous DNA free line which is to be imported in India has to be imported under the provisions of this particular act, right? Seeds Act 1966. What does the Seeds Act 1966 do? And now we have another updated seeds bill also coming up. So what you will do after genome editing? After genome editing, ultimately what is going to be your product? Seeds, right? So in the context which we are talking about is seeds. So here we are targeting to develop some high yielding seeds with better attributes. So selling the seed, developing and selling the seed comes under this particular act. So there is entire process of how a new variety is notified. Especially plant breeding students will know more about that how a new variety is notified. So any variety which is developed through genome editing will also go into the pipeline as prescribed under the Seeds Act. Insecticides Act 1968. Sometimes, for example, herbicide tolerant crops if we talk about. The other day also there was one example I showed that some non-GMO herbicide tolerant trait has been developed. But again, the herbicide which is going to be used on that particular crop is regulated under this act, Insecticides Act. So for example, if you say develop any, say for example, glyphosate resistant crop by some means, but is glyphosate allowed to be sprayed on that particular crop? Because in India under this particular law we have label claims that this particular insecticide or the pesticide is allowed to be used in this particular crop in this particular dosage. So we have a separate institutions about that, Central Insecticide Board, and we have many different institutions who do that. Then of course the Patents Act 1970, about which I am going to talk. So we have talked about the vectors, like in your hackathon also you have used the vectors, the 4 groups. But are those vectors freely usable? There are intellectual property implications about using those vectors, right? Even the technology of genome editing, there is an intellectual property, there's a big intellectual property even dispute going on between the 2 big groups, the Doudna group and the MIT group. Environment Protection Act 1986. So the SOPs are particularly from this particular act. So the standard operating process, which we have heard in the previous lecture, they all come from this Environment Protection Act 1986. So under Environment Protection Act, this legislation of regulating GM crops, GMOs, have been developed under Rules 1989. Then there is APEDA Act. So what was the idea of the Arjun group? Where they're working on the Basmati line? Okay. So for your information, you cannot work on Basmati line, because under this particular act, Agricultural and Processed Food Products Export Development Authority 1986, a notification has come in which there is a restriction on using Basmati varieties for genetic engineering. So when I was a master's student, even a PhD student, most of my colleagues who were working in rice, they would use Pusa Basmati 1 as their genotype of choice to do plant transformation. And somehow it was told to me that it's a very good genotype for transformation purpose. But then this notification came and everybody stopped working on Pusa Basmati 1 and other different rice varieties, MTU 1010 and others were adopted. So Arjun Group, as soon as you give this information in the form D1 to your IBSC, the IBSC will immediately turn down your proposal that the work which you are proposing cannot be done in the Basmati rice lines because of this particular restriction from this act, right? So but now you know, right? So this is important. So many times what happens, people do things and then they come to know that okay, it was not legally permissible, it was unlawful. And for example, when I was a PhD student, At that point of time, although I was in ICGB, it was an international organization. So you used to send a lot of biological materials to different countries and receive material from different countries. For example, my thesis work started when I received some particular construct from Max Planck Institute of Developmental Biology, Tübingen, Germany. But I didn't know at that point of time that whether we get to have some legal permit for that or not. Similarly, we used to send a lot of samples for sequencing, for example, plasmid sequences. At that point of time, sequencing facilities was not very developed in India, so we used to send to South Korea. Microgen, I think that company still takes the samples. And also we used to send a lot of our colleagues who are in all over the world, so we will get requests, okay, send me that strain. For example, somebody is in a sandwich PhD program, half work done in ICGB, half work going somewhere else, and then we will get a request, okay, please send some stab cultures of yeast to us in America. So we used to do FedEx, right? But those things is not permitted. We cannot send any biological material outside India without taking permission under a particular act, which is the number 9, Biological Diversity Act 2002. Any genetic organism or any of the biodiversity cannot be sent outside India without taking permission from this authority, which is in Chennai. So in this training program, it is our duty to tell you that all the steps one has to do in a lawful manner only, right? Genome editing should be done in a lawful manner, and these 11 laws are there which affect different steps of genome editing. Getting a construct, getting some line, giving some construct, the intellectual property behind that, the quarantine regulations, different aspects are there. So this was to give you some flavor that there is something, not in detail. So basmati rice is definitely out of question under this APEDA Act. Then we have GI. So geographical indications, you must have heard that in different parts, for example, even basmati is a GI product, and we have similar GI products in different parts of our country. Now, if you want to do genome editing in the GI commodities, then this will have, you have to really check whether you can change that, right? Then PPVFRA, this is the most important act which will be of very much significance for genome editing, and I will talk about this in bit detail in the coming slides. Biological Diversity Act, I mentioned, you can send any material outside India without taking permission. Then Food Safety and Standards Act 2006. So this Environment Protection Act, this is for environmental release. For example, if you want to release something in the environment, so this act comes into force. I'm talking about genetic product. I'm not talking about the SDN1 exogenous free DNA. Although they have also been notified to be exempted from Food Safety and Standards Act. Okay, so any food crop, for example, okay, you got everything right and you got an environmental clearance to grow, for example, genome edited or transgenic rice, but it's a food crop. So if a crop is a food crop, then one has to get a specific permission from Food Safety and Standards Authority of India. So anything food also comes under this particular act. Then Legal Metrology Act 2009. This is, many people don't know that in India we have a very strong labeling law. This is another area, you know, if you will follow the developments in European countries, so they are very strict about the labels, the labeling law. For example, you go to a superstore, pick up something, and what are the ingredients inside it, what should be written. Anything you pick, so many things are written. Their nutritional composition, weight, use by date. So why it is written? Because everything has been notified under Legal Metrology Act that what a packet should contain. This act under one notification has also notified that if any package contains the GMOs, then on the principal display of that package the word GM should be written. For example, if you go to the supermarket or anywhere, you see that for vegetarian you see that green dot sign, for egg the brown, for the meat products you have red. So why they have? Because of this particular act only. And this act also says that if any package contains a GM, that GM word should also be mentioned. So, and all these laws actually, from where they are coming from? So they are coming from many international conventions and treaties. So I'm not going into detail about that. But for example, this International Plant Protection Convention, 185 countries have signed that convention, India being one of them. So now this is a global aspect. So the laws which I have mentioned were India specific. The laws are territorial. Our parliament has made the law. Our parliament has a jurisdiction to make laws only for India, not for outside India. So every country have their own laws. So most of the laws, it is expected that the signing parties will harmonize the law, right? But sometimes it is harmonized, sometimes it is not harmonized. For example, if you are a PhD holder, also a science and genome editing and everything, and you are recruited in a multinational company in their regulatory affairs department, for example, you are the general manager regulatory affairs of this Inc. company, then you have to know about these laws in many different countries, because multinational companies operate in multiple jurisdictions. So like this International Treaty on Plant Genetic Resources for Food and Agriculture, so our that genetic law, that PPVFRA law is harmonized with this. Then WTO agreement, the TRIPS agreement, the International Convention for the Protection of New Varieties, so this PPVFRA Act, Convention on Biological Diversity, so that the Chennai authority is here, Cartagena Protocol on Biosafety, Codex Alimentarius, right? So these are all the international treaties, and India is a party of all of them. So as a PhD student, as a trainee of genome editing, some indication you must have that, okay, there is a global landscape, and you know, all this research, product development happens under this global landscape and also the national legal framework. So now let's come to the IP landscape of genome editing. Specifically now we are focusing on the intellectual property, that is the Patents Act 1970. So what is a patent? Patent is an exclusive right. It is given, is an exclusive right granted for an invention which is a product or a process that provides in general a new way of doing something or offers a new technical solution to a problem. To get a patent, technical information about the invention must be disclosed in the public. So what is patent? It's an exclusive right, and in lieu of what? In lieu of public disclosure. For example, you have done some innovation as a scientist. Now you are applying for a patent. So what does it mean? It means that you are disclosing whatever you have developed, the process which you have developed or the steps you have used in your innovation. You are disclosing it to the general public. And why this disclosure is important? So that other people can work on that, they can improve it further, right? And but what you are getting in by disclosing it, you are getting some protection that, okay, for next 20 years you have the sole rights of licensing this technology, right? So it is a give and take. I disclose my invention, and in return what I get is protection for 20 years. So essentially patent is this. So in India, the Patent Act 1970, and there are different kinds of amendments, and these comes on and on. So when you are in the regulatory affairs department, of course you will be keeping in touch with all of them. So in your company, for example, when I was in a company, so every week at that point of time, this IBSC and all, this RCGM and other committees used to meet. So every week you used to get an email from our regulatory affairs department that this is the development which has happened in different laws or amendments which have come. So criteria of patentability, I'm just these probably revision for you, that it should be novel, it should have an inventive step and must be non-obvious, it should be capable of industrial application, and it should not attract the provisions of section 3 and 4 of the Patents Act 1970. So this is important for genome editing. So let's see why it is important for genome editing. So what are those section 3 and 4 of the Patents Act 1970? It says that a method of agriculture or horticulture. So it is talking about what cannot be patented, right? So everything can be patented, but there are some restrictions which cannot be patented. A method of agriculture and horticulture cannot be patented. For example, if you do some agronomic research and you come up with the technology that, okay, by doing this row to row distance or by doing this kinds of planting geometry, I can enhance the yield and I can do this, I can do that. Please give me a patent. No. Any method of agriculture, horticulture, no patent. I have developed a new technology of grafting in such a way that you can have oranges as well as apples and mango in the same tree. A very novel method of grafting in which my guide RNA is coming from the rootstock and doing this in this. Patent? No. Any method of agriculture, horticulture cannot be patented. So unnecessary, don't like filing application. If it is a method of agriculture, horticulture, no. Then plants and animals in whole or any part thereof, other than microorganisms but including seeds, varieties and species, and essentially biological processes for production or propagation of plants and animals, or any living thing in nature. So under the Patent Act, these things are not patentable. And any part thereof, so any plant or any part of the plant cannot be patented. Right? So this is a plant. Now the question comes with us for the genome edited plant. Like for after 5 years of work and after investing 3 crores of money, you have developed a plant. At the end of the day, what is in your hand is a plant. So can you patent that plant? No, you cannot patent the plant. Similarly, if it is any animal, you cannot patent that animal. Either plant, animal, any living creature cannot be patented. Also any part thereof. So this is very important because this will be a matter of discussion in a court case, that what is a part of a plant. For example, you have used this genome editing and you have developed a construct and everything, and then your plant has got a mutation, a very good mutation. Yield is increased by 50%. Okay, but now that mutation is where? It is in the DNA. And where is the DNA? It is inside the plant. In the plant cell, there is a nucleus in which that DNA is there. So is your mutation a part of the plant? That is a question here. So after the genome editing, whatever we are developing, if it is a part of the plant, which apparently looks like because it is a part of the plant, then it has to be dealt in that manner, which means that it cannot be patented. Then let's talk about gene patents and a global thing, that a gene patent is the exclusive right to a specific sequence of DNA. For example, DNA sequences cannot be patented even under US law. So human genes cannot be patented. There is a US Supreme Court decision on that because DNA is a product of nature. Even in US, DNA as such cannot be patented. But DNA manipulated in lab is eligible to be patented. If any sequence which does not exist in nature as such, right? So now the science comes into that. For example, okay, all the scientists want to develop innovations and get intellectual property, but DNA cannot be patented. Then what can be patented? If DNA is manipulated in the laboratory, it can be patented. So for example, there is a gene. The gene cannot be patented, but its cDNA can be patented, right? Because cDNA cannot be, it doesn't exist in nature. The cDNA has to be created in the laboratory, right? So similarly, the recombinant DNA and plasmids, they are under patent. So now, our things start. So this is clear. If anything becomes a part of the plant, so it has to be dealt as a part of the plant. DNA sequences as such cannot be patented, right? But genes and constructs can be patented. And that is what has happened in the CRISPR-Cas9 technology also. So another thing is, as I told, that genes cannot be patented. So now tell me whether CRISPR can be patented. What is CRISPR? These are some sequences of bacterial origin, right? So CRISPR cannot be patented. And now you are having a lecture about the intellectual property of CRISPR-Cas9. And what about Cas9? Cas9 is also a naturally occurring protein from somewhere, sp-Cas9 obtained from an organism. So even Cas9 cannot be patented. Neither CRISPR can be patented, nor Cas9 can be patented. Then why we are having this lecture? Right? So neither CRISPR can be patented, nor Cas9 can be patented. They are naturally occurring protein and partly naturally occurring bacterial processes. So what all these patents, what they have done, so these big patents, these parties have sought to patent methods, engineered components and compositions to target specific genes and achieve specific effects. So if somebody says that okay, CRISPR is under patent, what you will do? CRISPR is not under patent. Cas9 is not under the patent, but different kinds of modifications of that, cDNA kind of modifications, the constructs, the plasmids, those things are under patent. So we also have another lecture about the IP landscape. Dr. Vibha Dhawan has been invited, so probably she will throw more light on this. So I am not going into depth of this. But there are two foundational group of patents. One is the UC Berkeley, where Doudna and other people worked, and Broad Institute, Dr. Zhang, who showed it first in the eukaryotes. So outcome, and there is a court case going on, not only in US, but in different countries. So the outcome will differ from country to country. And there are many different kinds of patents, about 11,000 patent families for CRISPR-Cas9 if you search in the literature. Corteva Agrosciences, they have taken all the patents from the MIT group, and they are giving licenses like the research license. So you will be surprised to know that even for research one has to take the research license. We are not talking about the product development. Even to do research one has to take a research license, and many organizations do take research license. And we are also in ICR, for example, now we have started working on genome editing in a big way. So we are also discussing about this, that whether or not and how and at what stage one has to take license, whether one has to take the research license or the commercial license. So this part I think I will skip about the global trends in CRISPR patent filing. But what I will come quickly is that in India also these groups have filed their patent claims. So it's not like this that okay, see, I already told that all the laws are country specific. So okay, they might be having some court case in America, but let's do it in India. No, they have also filed, both the groups have filed for their patent claims in Indian Patent Office as well. So this, like everybody gets vectors from ad gene to start the work. But are the ad genes vector free of intellectual property? No, they are not. So no implied license is there. So I am not going to read it, but the gist of this is that ad gene is just a repository. So there is a depositing scientist and there is a requesting scientist, and the plasmid is deposited and plasmid is requested, and a MTA, the material transfer agreement, is signed. But this MTA doesn't mean that after signing the MTA you have got the license to use, right? It is the material transfer agreement only. The implicit intellectual property still it retains, the plasmid retains the intellectual property which are embedded in it, and for that one has to enter into separate negotiations and separate licensing agreement. But I think I will stop here by telling you one thing, and this has become very much of a point of discussion in India, and this has become a point of discussion in India after this BT cotton. So first we have this BT cotton 1, that is a Bolgard 1, and then came the Bolgard 2. So I do not have time to tell you the entire story behind that, but this thing has been debated through and through in Delhi High Court. And then this point came up that okay, for example, in BT cotton, the court case was between Nuzividu Seeds, Hyderabad, and Monsanto Maheko Biotech Limited. So both of these parties, so the Mahiko Monsanto party, they are giving license to many different seed companies. So now what the Nuzividu lawyers claim, they say that, okay, you have developed this very good construct, and you have also developed this very good BT gene, it confers protection, everything fine. But the moment you have developed the transgenic, the moment the BT gene has been incorporated in the plant, it has become the part of the plant. So if it has become the part of the plant, then why you are bringing me to this court under the Patents Act? Because the part of the plant you cannot patent. So what Nuzivudi Group said, that at most what you can do is you can apply for PPVFRA protection. So we have another, that PPVFRA, it provides protection to the varieties. So they said their logic was this, that once any innovation becomes part of the plant, it cannot be regulated under the Patents Act, and it should be regulated under PPVFRA Act. So in genome editing also, as soon as all these things will come, so what is going to happen is that the nucleic acid sequence is a chemical product. It's not capable of industrial application until it is first integrated into a plant cell, where it can express itself through essentially biological process of transcription, translation, replication, right? So, okay, guide RNA you have designed by a great innovation, you have created the mutation, but once it becomes a part of the plant, then what happens is everything happens as a variety only. So as our country will commercialize these products, we will see that how these are regulated, especially the intellectual property aspect is concerned, right? So present understanding is that genome edited variety can be released as EDVs, essentially derived varieties. Many of our plant breeding students will know this term, EDVs, and what does it mean, right? So for example, MTU 1010, many people are doing genome editing, but it's already a variety protected under PPVFRA Act. If we change one particular locus by doing genome editing, it becomes an EDV. So this part I'm not talking about the implications of the Environment Protection Act, just that again this also has got so many different kinds of guidelines and all. So this CRISPR-Cas9 genome editing, unless it becomes exogenous DNA free and certified to be such, till that date everything is a transgenic research. So you as a student, when you will be working in your laboratories, so don't be under this impression that I'm working in a new breeding technology or genome editing, so this is transgene free. No. For most of your PhD work or whatever work, it will be a transgenic thing. It will be a recombinant thing. So all the regulations, all the guidelines, all these 23 guidelines which are mentioned under Environment Protection Act will apply to these plants also. Only when the exogenous DNA free status is confirmed by the IBSC, then that exemption from rule number 9 to 11 is given. So this part has been told already, that form D1. So even before you start research, you make sure that you apply in this particular format. Like for example, in our laboratory we are going to start working on some new genes. So now I am going to fill up this form D1, specifying that this is the thing which I am going to do. So again, import and export or even exchange, getting plasmid from one laboratory to another laboratory, at least recombinant DNA movement is not free. You cannot give a plasmid to your colleague somewhere and get a recombinant plasmid from your colleague from some other institution. So there is a process, because it's again the plasmid is a recombinant DNA product, right? So how much, all these things have been specified. So it's कहते हैं ना कि हर चीज लिखी हुई है. So here also everything is codified, it's written there. If it is this milligram or this nanogram, then what will be the process? So I'm not bombarding with you all that detail, just sensitizing you that this area has got all these legal aspects. So plant quarantine I was talking about. So this is one real life case study when I imported some of the germplasm from US. So what I did, so I was the importer and from here I got the material. So I got this permit. So import permit, and this is for non-GMO. For GMO also I got separate permit, but for non-GMO also got a permit for that. So this is also is another story. So everything has to be done properly under legal framework. So very good. We are very excited about genome editing, but it is also a duty to tell you that to do genome editing in a lawful way, because genome editing being a technology is an offshoot of recombinant DNA technology. So it is also under different kinds of regulations. Those 12 acts which I have shown, they all apply to genome edited crops, right from quarantine to intellectual property and whatnot. So this was for your sensitization purpose. So with this I will stop here, and if there are any questions I would like to take before we disperse for lunch. Yeah, see, any reagent which you order from Sigma or somewhere, even that if you read the fine label, so what is written there, that they are not giving you a license. Means if you are ordering something, a chemical or any construct or any kit even, so it doesn't mean that you have got the license for that. So here also actually this is again a question which has to be addressed at a higher level, whether to get license and then start the research. But most of the laboratories in India, they have started working on genome editing without the license, with the available technology and all. And the understanding is that as that time comes, whenever there is that time will come to take the license, at that point of time the license will be negotiated. Some institutions, for example, ICRISAT in Hyderabad, because they are an international organization, they want to do more probably in a systematic way, so they have taken the research license also. So it depends on your institution. As a researcher, your duty will probably stop at that point of time when you apply in the form D1, disclosing that this is what you are going to do. And now you are putting that application to your director or vice chancellor, and they are going to give you permission or to start that work. So once you got that permission from your institutional biosafety authority, so you are good to go. You need not wait that, okay, now I will get license, now I will get this and that. But yeah, reporting from your side should always be perfect. So you start your work with disclosure that I am going to do this particular work. And because most of you are students at this point of time, so your guide has to fill that particular form. And if somehow it misses, then it's your duty to remind that, sir or madam, should we also intimate our IBSC or not? Because in the SOP lecture what you got to know that, okay, once a product is developed, then all the data from day one is requested actually. Even those particular lines which are not exogenous DNA free, they also act as your positive control. So it's not like this that you are going to throw away all the material. Any other question? Yes.
So many scientists are registering clones, construct or plasmid as a patent. So there is a chance of Copyright Act also?
Copyright Act?
Yes, sir.
For example, these softwares,
Yes.
like the guide RNA designing softwares, Chop or whatever, even if you design something, and also Dr. Soham Ray also mentioned about one of the software which was developed here in IRI, PTG assembler. So we have got copyright for that, right? So all the softwares are generally given protection under copyright.
Okay, sir.
But any software which is protected under copyright doesn't mean that if you use that software to design a guide RNA or something, then you need the permission of the copyright holder. No, it's not that case. Guide RNA can be designed without taking permission from the copyright holder. Copyright only applies that you cannot make copies of that software and start selling the copies of that software in some CD, or you can put it in your own website. For example, whatever the algorithm is behind Chop Chop. Okay, so now you are not supposed to use that particular code without taking permission from the copyright holder. So copyright is relatively simpler, it stops there itself, just for the distribution of the software.
Thank you, sir.
Okay, thank you. So lunch time? Okay, thank you. हाँ लंच के बाद है देखते हैं I have already explained these questions, तो अभी बंद कर सकते हैं इसको. नहीं अभी नहीं होगा. We are going for lunch now. लंच के बाद देख लेते हैं क्या है. यहीं पे है
प्रेजेंटेशन
2 से 3:15 सैंडल सीक्वेंसिंग. तो ये जो है सैंडल सीक्वेंसिंग तो कुछ प्रेजेंटेशन तो होगा, कुछ पावरपॉइंट पर. So after lunch everybody will come back to this auditorium. So next session after lunch will be here itself.
बोल दे, मैं यहाँ लगा देता हूँ.